training programs from ISTA authorized or ISTA itself organized training programs have attended. So I'll try to give you a overall perspective on ISTA and what about its testing methods like that. With this, I'll start my presentation. First, what is ISTA? We all know that the history of seed testing, it dates back to up to like mid of 19th century. That is, we all know that Frederick Nobe, we know that he is a father of seed testing. In 1869, he actually published his handbook on seed testing. From that point onwards, this interest on seed testing, that means the knowledge on seed testing, it started itself unraveling across the world. Means people tried, gave more emphasis on quality of the seed. Means quality of the seed is how much important for getting the right yield. They started noticing the importance of seed. So they started giving emphasis on the testing aspects. Means how to test whether the seed is right or wrong. From that point onwards, slowly, when we come to this first of this 20th century, the dominant stakeholders, they used to organize this international seed testing congresses and this thing. That is the first point how ISTA got its origin. That means in first congress which got held in Germany, the second one in Netherlands, third one in Denmark, and the fourth one, the fourth International Seed Testing Congress, which got organized in UK, was the basis for this setup of this International Seed Testing Association, which you can see from the slide that in 1924, 26 countries participated in this thing, and it took the shape of an association in 1924. Okay. So then what is ISTA? So ISTA is an international non-profit association. It is an association of seed testing laboratories and an association of individual seed professionals also. That means you can join it, this thing as a personal member also. So who operates ISTA means ISTA is operated by an this thing executive council, which means who that council means it is by the it is under the governance of the government of the member countries. That means each member country will have a casting vote. They actually elect the members of this executive council. Apart from that, other technical committees also will be there. They will be purely on this thing basis. That means people can voluntarily enter into that this thing, depending upon their research background. But they are not this thing. They only ISTA executive council only is the paid and actually executive. It will only deploy its executive functions through this executive council, which has around 11 members. The current ISTA president is from India only, Dr. Keshalu sir. He is director of Telangana State Seed and Organic Certification Agency. So this is in brief about ISTA. What is ISTA? It is a non-profit association, which is basically formed because of the association of seed testing laboratories and individual professionals, seed professionals. Okay. Then what is the basic vision of ISTA means? Its vision is to have this thing, testing in seed testing. It should be of whatever the participating laboratories are there. They should have a uniform method of testing. That is actually the ISTA vision. That means whatever the laboratories they are following, they all should follow the same standard procedures, so they can generate reproducible results, which will help in seed trade and for getting the actual potential of the seed material. That is their basic motto. That means uniformity in testing they should achieve. That's why they want to develop some of the protocols, and these protocols are being followed uniformly, and they are mandatorily be followed in ISTA accredited laboratories. Other laboratories also, if they want to line up their proficiency in tune to this excelling laboratories, they have to follow this testing methodologies only. So basically, ISTA rules means what? These are nothing but the testing methodologies which has been developed, which has been adapted across these member laboratories to have a reliable and reproducible result. That is the basis of ISTA rule. ISTA rule is nothing but the methodology. It is not the it is not the range. It is just the methodology. ISTA rules are, it indicates the methods to which we have to follow for doing a particular test. For example, we take the germination test. Germination test means how to conduct a test, how to take a which substrate has to be taken for which species, which temperature has to be taken, what are the dormancy elevation treatments has to be done, what is the first count, what is the second count. That means what is the detailed methodology that has to be followed, that will be given in the form of different chapters. That is what ISTA rules means. ISTA rules, normally the basic misconception is that they will give the upper and lower limits, like your IMSCS, like Indian minimum seed certification standards. Now, for example, in case of paddy, the germination percentage should be this much to have it as a good quality seed. But in other crop, the germination percentage should be more than 85% or more than 90% or more than 95%. It is not that one. It indicates the methodology to do that particular test. ISTA rules means it indicates the methodology to perform that particular test in an acceptable and reproducible manner. That is the basic this thing which you have to get from that nomenclature. So with this background, what is the primary aim, which I have already mentioned, that the primary aim of the ISTA rules is to provide testing methods for seeds designated for growing crops or production of plants. Then ISTA, from where we already seen when it got started, that is in 1924, it got its this thing. From that point onwards, the methods the methods have been proposed. They are being validated across its laboratories and are being followed in its accredited laboratories. Okay. So with respect to its presence, it is spanned in more than 83 countries. And in case of India, it was almost 15 member labs are there. Our own ICR NISST, our lab is a member lab, not an accredited lab. So I'll let you know what the difference between a member lab and accredited lab. With respect to accredited labs in India, we have 9 accredited labs. The recent one might be this one, East West and Advanta sites. Out of these 9 accredited labs, 7 are from private sector and 2 are from government, and the 2 are, one is from this Tamil Nadu State Seed Certification Lab, and the other one is the Telangana State Seed Certification and Organic State Seed and Organic Certification Agency Lab. Both these 2 public sector labs were accredited labs in India, whereas in worldwide more than 130 accredited labs are there. And if you see member laboratories, it is almost roughly reaching to 230 member labs are there worldwide. So they have a very broad presence across the globe. Why? Because of the proficiency in testing, because the methodology, what they are being suggesting, and how it is being validated rigorously, and how it is being published, and how it is being professed. All these parameters actually gave that this iconic status basically for this ISTA itself. For example, if a particular seed lot is carrying this ISTA certificate, means worldwide, the importing country will happily accept that seed lot. It means they are assured of the quality of that particular seed lot. That means whatever being, if the lot is being carried with either with orange seed lot certificate or blue seed sample certificate, means that like the lot is of very good quality. That means they have gained that this thing because of the methodology that what they are being followed, and how rigorously they are being implemented across its accredited laboratories. That's why that this thing is there for this ISTA methodology itself. ISTA rules are nothing but encompassing all these methods. If we see this thing of ISTA, we can see one part is already here. We mentioned that this thing, one is ISTA rules, that is the right this thing. The top one is issuance of orange and blue certificate. That means whether the orange certificate is for seed lot certificate basically, and blue is for a seed sample certificate. That I'll explain a little bit later. And the other part of this thing is for this publication aspect of scientific results. So many of you might be knowing that ISTA has its own journal like Seed Science and Technology, which is having around 7 NAS rating. So like that, this thing. Apart from the fourth wing is to this thing evaluate the laboratory performance, whether the member lab is proficient enough to grant accreditation or not. That is also one of the basic fortitude of this ISTA. So all these 4 broad functions ISTA normally carries out. So this is with respect to ISTA. I think some of the students might be from seed technology background also. So that's what, if you are having any questions that this thing, you can ask me at last. I'll try to, if I want this thing also, I'll try to get back to you by knowing that this thing. Okay. This is broadly with respect to the ISTA as such. And I just want to highlight one thing here, that it is more of a, rather than here, nothing much to explain. It is more of a like a book sort of thing. I mean, this means this ISTA rules are having 19 chapters are there, out of which 17 chapters deal with in particular to particular tests. For example, like after sampling, what is the basic test? Purity test. After purity, they will go for measurement of other seeds. Then your germination test. Then your for sake of viability, for sake of vigor, for sake of embryo excision, for sake of GMOs. Like that, each chapter will deal about a particular this thing. So ISTA rules it itself span around like roughly some 320 to 330 pages. So ISTA rules will know means we will know the entire range of seed science we are basically covering. If ISTA rules is that much broad topic, that means each topic itself is so huge. ISTA rules, we are just, this thing means we are actually covering the whole aspect of this thing itself, the testing part of entire seed technology subject itself. Okay. So with respect to that ISTA rules, it is basically the methodologies where what they basically describe. For example, we'll take an example of a particular test, germination test. So what is the object of germination test? To evaluate the germination percentage of that particular lot. Then definitions. Definitions means in case of germination, what are the things we know? That normal seedlings, abnormal seedlings, hard seeds, dead seeds, fresh ungerminated seeds. So they will give the definitions of all these things. Then for sake of general principles, then means how to conduct a, means what are the basic things which we have to follow for doing a particular test, what are the operators, and how what is the workflow of the test, how we will express these results, and what are the tolerances that can be permitted within a particular test. All these things will be described in that particular chapter. For example, each chapter comprises one particular this thing aspect, right from sampling onwards, so like the physical purity, germination, other seed determination, like that. Each chapter encompasses one this thing. So ISTA rules, they are broadly designated for almost all crop species. For example, agriculture, vegetable, tree, shrub, flowers, spice, herb, medicinal, all these things it broadly categorizes. Whereas with respect to ISTA rules, whether they are like sacrosanct and they will be restricted to particular set, means no. You can suggest for a particular species, if for a particular species, for example, a medicinal plant. For a particular medicinal plant, if ISTA methodology is not there, you can propose it for inclusion. That this thing. That means you can represent it to ISTA secretariat. Then they will take a call whether first they will submit that your proposal to ISTA technical committees. For example, you are referring it for a germination test. Then a particular committee, technical committee responsible for germination, they will evaluate your this thing. Then if it is fit for test, this thing means then they will do proper validation across multiple laboratories. Then with the results, they will submit it to ISTA Executive Council. Then ISTA Executive Council, then they will submit the same for General Assembly for voting. Once voting is done in favorable to particular inclusion, then that will be included in the next year coming ISTA rules. For example, this ISTA rules, it got started in 1924. If you see of 2025 also, compared to last year, compared to 2024, some of the things for some species or some methodology deviation for particular test, they will the amendments will be there first itself. So it is an evolving aspect. It is an evolving aspect with respect to this ISTA rules. So continuously it got means new species will get added, and the methodology will also get it to be streamlined by this thing. That's why they could able to maintain their this thing stance. That's why their methodologies are that much proficient, and everybody wants to adopt their methodologies. That this thing. We also, as per our this thing, we broadly follow ISTA methodology only. But IMSCS means it indicates only the limits. It is not the methodology. IMSCS is only for limits. Okay, we will basically follow ISTA methodology. Whatever the quality manual which are being followed either in certification agencies or law enforcement. That means this professing seed inspectors. They basically follow that this thing only. Okay. This thing testing manual only, which is broadly taken the crux from ISTA rules only. Okay, there are some variances will be there, but the basic soul is taken from ISTA only. Okay. So with respect to accreditation, what is I just want to give a brief about accreditation also. So what is accreditation? That means ISTA rules, now you have a decent understanding. Oh, this is a problem with online this thing, because I can't able to get the feedback immediately. So ISTA rules, I think most of you understand. That is ISTA rules means that is nothing but methodology to carry out specific seed testing procedure. Seed test normally what we will do seed testing on seed quality attributes. Either it might be physical purity or your germination or your moisture or your seed health aspects or any other special tests like your tetrasolium or your embryo excision or your for GMOs or like that. So all these things, if you are doing that, this thing is that methodology actually comprises of ISTA rules. Whereas with respect to how to become a ISTA accredited laboratory means, that means accreditation means that your laboratory is proficient enough to carry out a particular test as per ISTA rules. That means then they will grant you accreditation. First, your laboratory should be proficient enough both in terms of your technical expertise and in terms of the apparatus that is required to carry out that particular test. For example, I'll give you an example. In case of sampling, so for example, your dividers, if they are of particular this thing means their error is within the tolerance limit, then only that particular equipment can be designated as fit for purpose for doing that particular work. So both your equipment and your technical expertise should be in tune to the level of conducting this proficiency testing, which is being given by ISTA. For example, so what is the basic steps to get it? ISTA accreditation means like in case of Indian, that means first we have to become a member lab. Like our lab, getting a member lab is easy. That means just we have to pay the designated amount in Swiss francs to that ISTA head office. Then we will get ISTA membership. It is just giving money and purchasing. ISTA membership is like that. This thing, for example, it is now roughly like almost 4.5 lakh per year. So you pay the money, you can become ISTA member lab. After getting ISTA membership, the next point is to participation. You have to participate in proficiency testing. Your laboratory no need to participate in all the testing zones. For example, right from sampling, germination, right from sampling, purity analysis, germination testing, your viability testing, your moisture testing, your for seed health parameters, your species and variety testing for GMOs. So like that, so many are there. No need to take all. If you are this thing comfortable for only this physical purity and germination, but if you are taking germination, physical purity also you have to take, because for germination you will use only pure seed fraction of purity analysis. Okay, for germination you have to use only pure seed. So you have to invariably participate in physical purity this thing also. That means you can participate whatever the test. For example, if my lab is, we are confident enough to participate in physical purity germination, moisture. We will apply those these things for a species and variety testing. For GMOs, right now we don't require, no need to participate in that one. So you have, we will participate in that particular this thing, and we will try to for example, we will submit the result back to the team. Then they will have the original result. They will see the tolerance limits, and they will see our test result is matching with their test result by applying the tolerances. If your test result is matching with their test result, for example, germination, your their germination percentage is, for example, 85%. Your germination percentage is like 84% or something like that. And if it is within the tolerance for that particular species, then they will just give you A or A grade, like that. A, B, C, and this thing will be there. Okay. So if you get consecutively for 2 years A or B, then your laboratory is proficient enough in terms of technical expertise to do that particular test as per ISTA rules. Okay. Now we have to see about your quality management system. Quality management system means in terms of your apparatus, whether your apparatus is as per the quality standards prescribed by ISTA or not, and how actually you calibrated, and the quality manual. That means the SOPs, the standard operating procedure for a particular instrument. That means you take any basic instrument. That means who will be operating, how it will be operating, and periodically within how much time it will be the maintenance is being done, and those things has to be properly documented. That means both your technical expertise also will be evaluated by proficiency. Then quality management system means whether your laboratory in terms of your the equipment and how your quality manual SOP, whether they are in tune with the ISTA guidelines or not, they will check. How they will check? They will check through this audit. Okay. Audit means after you continuously will get 2 years for successive years, or even A, B, or that also will do. Then an on-site assessment of this thing will be there. Then after that, granting of accreditation will be there. Once your laboratory is granted accreditation, means your laboratory will be accredited for a period of 3 years, within which again you have to participate and you have to get it re-accredited. The main this thing is the crux is getting a proficiency, passing in proficiency by having a sound understanding on ISTA rules, then making your quality manuals and your operators in tune with the ISTA this thing. Okay, these are the basic requisites if you want your lab to get it accredited. But one more thing I just want to this thing, it involves quite amount of money is also involved. That means I already told each year you have to pay 4.5 roughly for membership itself. Then for your ISTA audit, you have to invite audit party. For that almost 10 to 12 lakh. That this thing means it involves quite amount of money. But the thing is, once your laboratory is getting accredited, means then you can issue certificates. Either for your this thing or for your consignment or any private party, they can send, they can request you that you can you please sample and test our this thing, so that we will export these lots, like that. You can generate a huge amount of money by that this thing. Okay. So whatever that this thing money you are spending this accreditation process, when we are issuing this orange seed lot or blue seed sample certificates, you can get that this thing. You can get that money flowed back, that this thing. The basis is your quality standard will be upgraded into international domain. So that is a basic crux of accreditation. Okay. Then with respect to I have already mentioned this. What are this seed lot and seed sample certificates? One is orange certificate, orange international seed lot certificate, and blue international seed sample certificate. What is orange certificate means? If both the sampling and testing both are done by your laboratory, then you can issue orange certificate. But only the party is submitting a sample. That means you are not involved in sampling. The sample is submitted by the party itself. You are doing only testing on the sample submitted. Then your this thing, you can issue only blue seed sample certificate. Then that means one is seed lot certificate, other one is seed sample certificate. That means in orange seed lot certificate, sampling also will be done by authorized person. The person who is doing sampling, we call him as a sampler. And the authorized sampler only will do the sampling, and after that we will do the testing. So it is more stringent. That means orange certificate is more stringent and more valid compared to blue. Why? Because in blue the sample is being submitted by the party itself. Your lab is not in your accredited lab is not involved in sampling. Your lab is involved only in only testing. Okay, that's why orange is more validated compared to blue. Okay. But I took the assumption that a little bit background is there, but wherever some clarity is needed at the last, you please ask, because the topic is like that. So at every this thing I can't elaborate, but wherever I want to just inform, I try to give an emphasis on that one. Okay, so this is basic from the, if you see the flow, first how ISTA got formed, then what are the ISTA rules, then what is actually accreditation itself means, then what is the meaning of this orange and blue certificates I just dealt till now. Then that's what with respect to this proficiency testing, all ISTA accredited laboratories, they have mandatorily they have to participate. For non-accredited laboratories, means if member laboratories, they have to participate if they want to get it accredited. For re-accreditation, it takes 3 years. Okay. This is with respect to basically what itself is with respect to ISTA the rules, but ISTA rules as such implies the method of testing. Okay, ISTA rules, they will infer nothing but testing methodology only. Okay, so if we see the testing methodology, what is the prerequisite for testing? Sampling. Sampling is the prerequisite for testing. That means if your sampling is robust, then your testing result is reliable. Otherwise, if your sampling itself is flawed, then there is no sanctity for your testing result. Okay, so that's why the sampling was given a very pivotal this thing in case of this ISTA realm. So first is sampling, then your purity analysis, then germination testing, moisture testing. After that, so many these things are there, depending upon the need and this thing. So just I won't deal with all. I will deal slightly touch some of the testing methodologies also. Okay. So why this sampling is very much required? We can see that. What is the meaning of sample? By taking a sample, we are inferring the entire quality of the lot. You know that the lot quantity, it is in case of maize, it is up to 40 ton. In case of paddy, it is 20 ton. That means by taking a small sample, you are inferring the quality of a 20 ton lot or a 40 ton lot. You see in terms of the money aspect, how much money is involved. So your sampling should be that much robust, that much rigorous, so that you are justifying, or you are the customer who is taking that this thing is not being cheated. That means that's why ISTA certificates, they are having that much sanctity. That means once it is passed through ISTA accredited lab is giving a certificate, means the buyer is the buyer or importer is assured of a good quality of that this thing. If you imagine the price in case of horticulture crops, for example, in case of for example, even a 100 even a 50 kg or 100 kg of tomato seed, imagine the price of that seed. So that's what, so much economics is involved. Everything finally boils down to economics only here also. So when money is involved, the rigorous this thing should be at its peak. So that's why the sampling is given that much sanctity. That's why in orange seed lot certificate, the sampler from accredited laboratory only will go and take the sample. Okay, because that sampling, there is a huge chance that in sampling itself, you will make huge blunder. In case of Indian context, you see, whenever a seed certification agency or a seed inspector, they will just randomly put a hand in that this thing, randomly they will take. But it is not how you have to, you can take. If you want to have a representative portion, means there is a scientific methodology which you have to follow. The same thing which I want to keep, for example, in case of a paddy, the 20 ton lot paddy contains almost 1.25 billion seeds, out of which what is the submitted sample size? The submitted sample size is it should contain 25,000 seeds. The working sample size, it should contain 2,500 seeds. That means Out of for 20 ton, we are examining for case in case of paddy only 40 gram. For 20 ton, we are examining 40 gram means that much rigorous. In case of germination, only 400 seeds. That means one seed out of 31 lakh seed, we are just testing. So your sampling should be most rigorous. That's why in case of outside our this thing, the seed samplers, the person who is taking sample, that is one of the highest paid jobs. Okay, that much importance that they will be giving for sampling itself. Okay. So here I don't want to explain in detail. So means how this means this is one of the sampling equipment. How they will just this thing. How to insert here. The angle of insertion is also important. How to turn means when they will insert, they will insert with the whatever the appendage that is downwards, and they will put it in a particular 40-degree angle, and they will turn it, and they will try to collect that particular sample. I think some of you know how what is a primary sample, what is a composite sample. Primary sample means, for example, in a single action, how much quantity of sample you will take from a seed lot, that is we will call it as a primary sample. For example, here you can use an instrument, or you can use in some of the cases even a hand sampling can also be practiced. So in one particular this thing, how much sample you can take it out, that it actually comprises of your primary sample. When you mix some primary samples, your composite sample will be formed. From that composite sample, the amount of this thing which we will submit it to lab is called submitted sample. Okay, the submitted sample normally should contain 25,000 seeds. Okay, from that submitted sample in lab for doing a particular test, working sample will be taken. That working sample is comprised of 2,500 seeds per se, that this thing. So it is basically a chain. Okay, but the thing is, the starting point is sampling itself. So the I don't want to deal all the aspects, but there is so much this thing is involved. Even how an aperture should be there. For example, the shape of the aperture also matters. That means, but some of the shapes are not valid as per ISTA rules. Some of the triers, they are also not validated as per ISTA rules. Some of the dividing methods, these dividers, you might be knowing that. For example, to have two equal halves, now normally we will have these dividers. But in these dividers, how many shoots, means how many appendages each divider is having, to have a representative portion in both the ends, that also will matter. So that means it should have a minimum of 16 shoots. So the number of shoots, that also is having a huge bearing on which type of divider which you have to use. I have already told at the time of proficiency, after proficiency, you have to have your quality management system. That means your apparatus also should be fit for user, and the method which you are following also should be as per standard operating procedure. Okay, then these are some of other methods which are specific for particular crop species. So that is basically with respect to sampling. There are tolerances are also there. Heterogeneity testing is also there, which I don't want to deal in detail. So that's what. So sampling is very important. From properly sampled this thing only, we will further proceed for determination of other aspects. For example, after sampling, the logical this thing is physical purity. Physical purity means the seed is of a same kind or not. We will check it. And here we have to be careful. Pure seed implies to species, crop species only, not at varietal level. Okay. For varietal this thing delineation, some other this thing is there. Pure seed means it is at a crop species level only. For pure seed, for each crop species delineated TSDs are there. That means pure seed definitions are there. So as per that pure seed definition only, we have to see whether that is a pure seed component or not. Okay, that I'll just show in this thing. Normally purity analysis, what are the components means pure seed, other seed, inert matter. Whereas in Indian context, as per IMSCS, here other seeds is also again bifurcated into other crop seeds and weed seeds. Okay, here we have 4 components. Okay, like a pure seed, other crop seed, weed seed, inert matter. Here pure seed, other seeds, and inert matter. Basically, pure seed means the seed that is represented to the same stated kind. That means as a party, if I am telling it is a paddy distinct means the pure seed is of paddy distinct. Okay. We are not bothered about variety level. Variety level, it will be taken care at the ODVs. Okay. It is at the species level. Okay. Other seed means other crop seed and weed seed. Okay. Inert matter means which won't fall in the above two categories. So I have told that for each crop species, one pure seed definition is there. Depending upon that one, the pure seed. For example, in case of pulses, if a seed is having a small portion of testi attached, means it is also considered as a pure seed. It is not that entire this thing will be there. For example, here you see for conditions for pure seed definition for 11. It includes all those the above mentioned crop species for Sicer, Glycine, Phaseolus, Pisum, and these things. For the first criterion is seed should be of having a portion of testa attached, then a piece of seed larger than 1/2 the original size, then seeds and pieces of seed entirely without testa are regarded as inert matter. Like this. For each this thing, PSD is there. For in case of paddy means the caryopsis, even if it is without that husk also in paddy, it is pure seed only. Then we can context how we can keep it for germination. But there one more criterion is there, huskless seed. There it will get eliminated. Not with respect to pure seed. Even a naked caryopsis is also a pure seed in case of paddy. Okay. That is with respect to here also I didn't deal in detail on this thing. I just touched what are the basic fringe outlines on purity analysis as such this thing. Then with respect to germination, germination basically in terms of botanical perspective is a simple emergence of radicalized germination. But as a seed technology, this thing, you should have all essential structures. The essential structures is a root word. How to determine the essential structures? There lies the science. And these essential structures, they are not same for all. It will vary with the crop species. What are essential? It all depends upon the crop species which we are testing. Okay. If all the essential structures are there, if they could able to produce a satisfactory plant under favorable conditions, means we can call it as a germination. Okay. So this I just copied from ISTA itself. So under normal seedlings, that's why the spelling is like that in some parts. You can see normal seedlings with intact seeds, seedlings with slightly defects, secondary infections. Hard seeds means it is the question of physical dormancy. FU seeds, fresh ungerminated seeds means it is this thing of physiological dormancy. Abnormal seedlings means which the seedlings which are not having all the essential structures, dead seeds we all know. So normally as per ISTA germination, we will categorize on these things. But the main question is what are those essential structures? Okay, here I want to emphasize, I think very few people will be of having an understanding on this one. For knowing the essential structure, first you have to have an understanding on the seedling group, the crop species which it belongs. For example, in case of you take the example of rice itself again. Okay, here rice belongs to the seedling group of A1232. First you should know which seedling group it belongs. For example, in case of here I just tabulated, rice belongs to A1232. Here A is for your agriculture and horticulture species. Means each number is designative of particular this thing. First you should understand that one. Then only you can categorize it, whether it is a normal seedling or an abnormal seedling. That's why I told whether to know essential structure is there or not. That itself is a science in evaluating of in evaluation of in germination evaluation basically. Okay, here you take for case of rice, it is A1232. A means it is basically for agriculture and horticulture crop. One is basically for monocot. If one is there here, one is there, it is a monocot crop. 2 is for dicot, 3 is for conifer. Okay, then the next one is the type of germination, whether it is episial and whether it is episial germination or hyposial germination. Okay, why it matters? Episial germination and hyposial germination. It matters. Why? Because the epicotyle and hypocotyle will have a bearing in classifying it as a normal seedling or abnormal seedling. Okay, in case of rice, we know it is a hyposial germination. That's why it is 2. Okay, in case of the third one is how the shoot development is there. The third one is in case of paddy, it is an enclosed coleoptile will be there. That's why that number. The most important one, the fourth one, that is 1, 2, 3, 2. The last one, the last one is Even though primary route is not there, if secondary routes are developed properly, we have to classify it under normal seedling only. Okay, that is most important. For example, people will see that for a rice seedling, if a primary route is not well, this thing, they won't bother about secondary routes. So they will simply classify it under abnormal seedling. That's why their lab in proficiency testing continuously they will fail. Why? Because first we should understand how to deal with that particular crop. How to deal with particular crop, how we will know? First we have to know what are the things that were mentioned in the ISTA rules itself. Okay, what is a POC definition, how a particular test has to be conducted. So this is what the crux. Here I want to highlight how many people we know that for rice belongs to which group. Then what are the things which we have to evaluate. So all these things will matter when we do a testing, especially for proficiency testing. If you want to procure a grade A or at least B, then all these things will matter. Okay, to evaluate a particular lot, this much emphasis is given. It is not like just randomly take a seedling, it is right, it is wrong. It is not like that. Okay, so just this one I want to emphasize. So in every this thing, this thing is there. Okay, then for moisture content, it is a basic this thing only. There are not much there not much this thing is there for detailed explanation. Okay, but we will follow that constant oven method, like 103 degrees for 17 or for most of the species. But what are the species where 130 degrees can be followed? What are the species 103 is followed? Where coarse grinding is required? Where fine grinding is required? Where detailed this thing is given in ISTA rules. Okay, just before proceeding for a particular test, first we have to see that this particular cross species, how we have to, if moisture content is more than 16% means how the pre-drying has to be how pre-drying has to be done, what are the methodology that has to be followed, how whether grinding is required or not. Whether grinding is required means whether it is coarse or fine. Then which method has to be followed. All these things are there in this thing. Yeah, apart from that, moisture is having it's this thing in economic aspect also. For example, if a particular seed lot is having more moisture means automatically the weight is more, and the buyer is at loss because he is purchasing actually water instead of seed. So that's why for moisture is also given utmost importance, because it is having a huge economic perspective also, apart from quality aspect, because even at the quality level also moisture is having a bearing for respiration, your seed health status, and all these things. Okay, so all these parameters are interlinked. So for each this thing, for each even a small this thing, so much detailing is needed. How a test has to be followed, and particularly what tolerance level equipment we can this thing means the size of even the size of the boxes which we have to use, and what is the tolerance range which can be permitted for a particular instrument itself. So everything matters for delivering a reproducible result. That's why the proficiency testing is one of the difficult this thing for getting it passed. So, but the thing is, we should have a thorough understanding on rules itself. We will casually take on rules aspect, then we will never able to do a particular test in a scientific manner. So people casually pass like germination, everybody can do. It is not like that. In every small this thing, even for a moisture testing also, if you see the chapter itself, if you do a proper moisture testing means it itself is a huge task. That's why that much emphasis is given, and each test is given a full chapter, and that much detailing was given. So that is what I want to this thing. That means there is a proverb that a devil lies in detail. So the detailing is the most important aspect, and that is the underlining this thing aspect of ISTA rules of testing. Okay. This is the broad which I want to this thing, because I don't want to deal any particular aspect in toto, because it itself comprises the entire seed testing aspect. I just want to listen. If you want to have any specific queries, I want to have if I can able to means I will try to answer that this thing. Thank you, and thank you for the organizers for giving me this opportunity to share some of the some of the aspects of this basic testing. Thank you.
Thank you, Uday sir. Thanks for sharing valuable information and your time, sir.
Thank you.
Any questions from participants? Any clarifications and queries?
But sorry, if you are from of a different from not from C technology background, I am a little bit fast. I may be because I have to do like that only because it is a huge this thing. So I'll try to explain. Sir, please.
Hello. Good afternoon, sir.
Good afternoon, sir.
Myself,
Please.
Dr. R.C. Meena, sir.
Sir, please tell, sir.
I want to know about the both the procedure of accreditation and the NABL accreditation. What is the difference between